Thanks for being here. Right, my name is Marcel. I'll be talking about WhatsApp, which is a tool we've been developing over the last two and a half years, roughly, which can be used for read-based phasing. And maybe you've even attended Shilpa's talk, one of my collaborators, and she talked about one certain aspect of the program yesterday here at the conference. But I will be talking more about the general tool. So I guess you know this already, but very briefly, what is phasing? So assume you have called a few variants on your genome, and then you know the genotypes, but you do not really know how the chromosomes look like. That is, you do not know what the haplotypes are. And that is really sometimes really important. That is what you're interested in. You want to know the relationships, which alleles are on the same chromosome. And applications may be investigating component heterozygosity. For example, if you have two damaging mutations on a gene, then you may be interested to know whether those those damages are on the same copy of the gene, then you still have a working copy, or whether both copies are affected. So there are different methods for doing this. You can do this in a wet lab with molecular methods by physically separating the chromosomes, and I've heard that is very difficult to do. You can do genetic phasing, which means you inspect the genotypes in a pedigree and deduce the haplotypes by using the rules of Mendelian inheritance. And you can do population-based phasing, which uses regions of shared ancestry in a population, and you can do read-based phasing, what I will be talking about, which is you use reads that span at least two heterozygous variants. And this, by the way, is also called haplotype assembly. So, read-based phasing, again, let's assume we have a chromosome and you've called variants on it, and these variants are, I'm just showing the heterozygous variants here. And you know all the alleles, you know the genotypes, and you have lots of reads mapped to that chromosome. And in principle, the only thing that you would need to do is to find a bipartition of the reads into the two haplotypes, here shown in blue and red. And then you can read off the haplotypes just here on the bottom from the reads. Very simple, sounds very simple, but in the presence of phasing errors, it turns out that this is really a difficult problem. The way to pose it is usually by the weighted minimum error correction problem, and that turns out to be NP-hard. However, WhatsApp, our tool, does find exact solutions, and it finds them efficiently. It uses by using a fixed parameter tractable formulation of the problem. And the parameter in this case is the coverage, which is different from what other tools do. Other tools have as parameter the read length, which is a bit unfortunate because you want really long reads, and then if you have long reads, you don't want your algorithm to have an exponential runtime in the read length. So we can deal with long reads in WhatsApp. I'm not going to talk about much more about the algorithm itself because that has already been presented at Recom 2014, so a while back, and you see here the JCB journal version from 2015. I will talk about how we turned a proof of principle implementation, just to explore the concept, into a, let's say, nearly production quality tool, and why, of course, you should use it. So as evidence, I will tell you that we have made more than 800 commits to the repository since the initial proof of concept implementation, aimed at improving speed, accuracy, but most of all, usability of the program. So the rest of this talk is going to be me telling you why you should use WhatsApp if you have need for read-based phasing. So WhatsApp is easy to use. That's at least what I think. And well, that applies to you if you're familiar with the command line. And in the simplest case, you just have to write WhatsApp phase, then the name of your input VCF file and the name of your input BAM file. So the required input is therefore just a VCF, and you can have one sample in there, you can have multiple samples in there. The input Required is furthermore a BAM file, and you can also give it multiple BAM files if it so happens that your read data is distributed among multiple BAM files. And we are actually working on CRAM support, and that is nearly done and will be integrated in the near future. The only thing that you really have to take care of is that the metadata stored in the header of your BAM file is accurate, such that WhatsApp can, if you want to phase multiple samples, can find where in which BAM file it needs to look for the reads that belong to the specific sample that is being phased. So, as I just said, WhatsApp supports standard VCF input, but it also outputs VCF files. And this slide may be a bit boring because that's kind of obvious that that's what you should do. You should also output VCF files, but this is actually something that not many other tools do. So how does it look like? You have an input VCF file, in this case with two variants, and both variants are heterozygous, which we can see because the genotype field is set to 0/1, and WhatsApp will phase the variants, and then it will add a PS tag, which stands for phase set, to your VCF file, and this is actually standardized in the VCF specification, and it will also modify the genotype tag. So the PS tag is basically just a unique identifier telling you in which group of phased variants that particular variant is. So if the ID is the same, then they are in the same group. And the GT tag now tells you how the phasing actually is, so how the alleles look like, what the haplotype is, sorry. And we even support read-backed phasing compatible output, which is basically the same information encoded in a slightly different way. WhatsApp will probably work on your data too. We do support quite many sequencing technologies, and we have tested the program with Illumina paired end reads. We have tested this with mate pair data. And our big focus was, of course, on PacBio reads because these are long and give you longer haplotypes. And then you can also put in anything that I would call contigs, that is a DNA sequence from which you know that it comes from one single molecule. And under that heading, I would also put synthetic long reads, aka molecular data. So that also works. And in addition, you can actually combine any of those technologies, and WhatsApp will just do the right thing and improve the phasing. So here's a nice feature that is maybe small, but it gives you a nice picture to show you. So WhatsApp can tag reads in your input BAM file by the haplotype they belong to. So if you look at this screenshot from the IGV, then you have lots of reads, and you can kind of see that they belong to different haplotypes, and you have two variants here, right? And then you can do phasing with WhatsApp, and then you can tag the reads in addition, and then you get this nice picture, and you can actually color them in IGV, and then you can see, you can really visually distinguish the two haplotypes from each other. So we have first haplotype here and the second haplotype there, and you can see that the alternative alleles are visible in those reads. So you can see in this example, I was using PacBio reads, and they are quite error prone. And it turned out that working with PacBio data poses another challenge that we needed to solve. So for each variant that we look at, that we want to phase, and for each read that covers that variant, we do need to find out whether that read supports the reference or the alternative allele. That information is stored nowhere else. It's not output by the variant caller, and it's not part of the BAM file. So we do need to rediscover that information somehow. The original version of the program just inspected the alignment. So it looked at all the reads covering a certain variant. And you can see here, this read obviously supports the alternative allele, just as these others do. But you can maybe see already what the problem is. For error-prone reads, such as PacBio data, this doesn't work too well in some cases. So here, that A, I as a human would put that one position to the left, and then that read would also support the alternative allele. Another example down here, there's an insertion of an A, and that should actually also be a read that supports the alternative allele. But just looking at this column here, we cannot detect that. So, and the problem here is that the aligner doesn't know about the alternative allele. It is biased towards the reference. So what we actually do in WhatsApp now is that we realign the reads locally, which other people also do, of course. And we do that by simply extracting a little bit of read sequence around the variant 10 bases upstream, 10 bases downstream. And then we align this to the unchanged reference allele, also with a bit of context. And then we also align it to the reference, but where we have inserted the alternative allele. And then we look at the alignment scores, and then the allele with the best alignment score simply wins and gets reported as the one that the read supports. And it turns out that this really improves our results on PacBio. But it also gives us something else, because we can use the same technique to actually detect and therefore also phase not only SNVs, but also insertions, deletion, and MNPs, and basically anything that can be stored in a VCF file, except for structural variations. So this is really something that not many other tools can do as well. I wouldn't be telling you this if I wasn't convinced and I could prove it to you that WhatsApp is actually accurate. So we've done an evaluation with the genome in a bottle data set from a single individual. We used Illumina reads for variant calling, so we get highly accurate variant calls. And then we use PacBio reads for the same individual in order to do phasing. The tools we compared were WhatsApp, Hapcut, and readback phasing from the GATK. On the left, you see the performance, and on the x-axis, we have the error rate, note the logarithmic scaling of the axis, and on the y-axis, you have the fraction of unphased variants. So in a perfect case, we would be here in the left, in the bottom left. And what you can see is, so each dot is a different coverage, and what you can see is that WhatsApp is the most accurate tool at identical coverages, So now we come to the point where I will just briefly summarize what my colleague Shilpa already talked about yesterday. So WhatsApp can actually combine read-based and genetic phasing. Genetic phasing, a very brief example for that, you have a trio. The mother has genotype AA, father TT, and child AT. Then you can immediately deduce that the A must be on the maternal haplotype in the child and the T on the paternal one. And the problem with that is this works only if at least one of your genotypes is homozygous. So in those cases, you want to phase with reads as well. And WhatsApp can actually do that. And the way to do it is you just use WhatsApp in the normal way, but you provide in addition a pad file that basically just describes the relationships between your samples, and then WhatsApp will switch to the pedigree-based phasing mode, pedigree phasing mode in which it will combine read-based and genetic phasing, and it will use whichever information it can to achieve the best result at each position. This requires less coverage, it turns out, and is actually more accurate than just single individual phasing alone. So very briefly, the results for this. Similar picture as before, we have different coverages for a single individual. Again, we want to be on the bottom left. This is the performance for a single individual. And if you now switch on pedigree phasing for a family trio, you get much better phasing results at lower coverage, so you get more for your money. Finally, I believe that WhatsApp is also a good citizen in the world of bioinformatics tools, at least, and that's why you should use it. So it is actually open source software under the MIT license. The algorithm is published and peer-reviewed, and I wouldn't be saying this if this were the case for all the tools out there. Documentation exists and is continually being improved. I believe we have somewhat readable code. We use C++ for the time-critical parts. We use Python for the glue code, and we have lots of automated tests. So in summary, WhatsApp is an efficient implementation of a peer-reviewed and accurate phasing algorithm. It's easy to use, it's standard compliant, it supports VCF output, it does support heterogeneous data, its support for realignment improves accuracy on packed biodata quite significantly, and it also enables us phasing of indels and MNPs. So these are unique features which you cannot find in any other program combined. And last but not least, we also have a very nice logo. So thanks for your attention.